and I will wait a few seconds just to let people in. Okay, so I think we can begin with the introductions. Welcome everyone to the 2021 Susie Bayarri lecture. It's a great honor for me to chair this session, just because for one thing, it's a session that honors a prominent Bayesian statistician, Susie Bayarri. I had the fortune to meet her, and it's great to have this lecture in her honor. She was, for those of you who don't know Susie, I invite you to read her bio at the ISBA webpage, and also her contributions, her papers in Bayesian statistics. She was also an amazing mentor and an inspiration for many young researchers. And the Susie Bayarri lecture is delivered always at the ISBA World Meetings. This is actually our second Susie Bayarri lecture by an outstanding researcher under the age of 35. So this tells you a little bit already about our speaker today. And it's my great pleasure to introduce our honor speaker, Tamara Broderick. Tamara is associate professor in the Department of Electrical Engineering and Computer Science at MIT. Her research focuses on developing and analyzing models for scalable Bayesian machine learning. Tamara has been awarded a lot of important recognitions, and I won't even try to enumerate them because I want to keep the time for her talk. But just to mention a few, she was selected to the COPS Leadership Academy. She had career grants from the Office of Naval Research and the NSF. She had a Sloan Research Fellowship, as well as Google and Amazon Research Awards. Tamara is a very active member of the ISBA community. She was an incredible junior ISBA member. I remember those years. And she received the ISBA Lifetime Members Junior Researcher Award, and also the Savage Award for her outstanding PhD dissertation work. So it's really a pleasure to have her here. I also want to introduce our discussants. We have two discussants today. Professor Sylvia Richardson, who's the director of the MRC Biostatistics unit at the University of Cambridge, and Professor David Dunson. He's the Arts and Science Professor of Statistics and Mathematics at Duke University. So just to tell you a little bit about the logistics, the way this will work, Tamara will give her talk for about 55 minutes, and then we will go with the discussants. We'll have Sylvia first and David later. For those of you attending, you can pose questions at any time. I will ask you to please use the Q&A feature rather than the chat, because it's easier to monitor the questions like that, but I will be, again, monitoring both. So Tamara will have time to reply to the discussants and then to answer your questions at the end. So Tamara, the floor is yours. And I have to stop sharing.
Thanks for the extremely kind introduction. And thanks so much for the invitation to be here today. This is really meaningful to me. I think, Suchi said something similar in her talk. I've really enjoyed going to ISBA and being a part of the ISBA community for a long time now. And so this definitely means a lot. Let me just go ahead and get set up here. Okay, so again, as I said, this is a huge honor. It's incredible to follow in the footsteps also of Veronica and James from previous years. They're superstars. I want to give a big +1 to what Raquel was just saying about this Susie Bayarri tribute that's on the ISBA website. It's really fantastic. It really covers her amazing life and many, many achievements. So I'll hopefully encourage people also to check that out. I think this is just one example of what an incredible community ISBA is. I really feel very lucky to be a part of it, and I hope we all get back together in person sometime soon. Speaking of great communities, to the extent that I have had any success in this job, it's been because I've had such amazing students and postdocs to work with. And so before I go on to today's lecture, I really want to highlight some fantastic folks who have been in my group on this project. The first author, the amazing first author of the work I'll be focusing on today is my just graduated PhD student, Raj Agarwal. So if you have a chance to interact with him, make sure to congratulate him for this amazing milestone. This is also work with my previously graduated PhD student, Jonathan Huggins, who's now an assistant professor at Boston University, and my probably will graduate in the next year PhD student, Brian Tripp, and they're all really fantastic. Okay, great. So let's get to the heart of the matter today. So we often want to find a sparse, interpretable set of covariates associated with the response, but interactions between covariates can make this a much harder problem. And so today we're going to talk about a fast and accurate solution. And as I go on, I just want to point out that, as Raquel said, we'll be taking questions later. If you have clarifying questions or questions that come up along the way, feel very free to ask them, and I'll try to cover them as well. Okay, so let's talk about a sort of motivating cartoon for why we wanted to solve this problem. Let's imagine that we have a bunch of gene expression data over, so say many genes. This could be hundreds of genes, thousands, millions. And we have collected data on a variety of people, say capital N people. And maybe in fact, we don't have data just on their gene expression levels, but we have data on various environmental factors, like how much are they smoking, how much are they drinking, how much vegetables are they eating. And we're interested in how this is associated with some health outcome, like maybe blood pressure level. Now, a typical thing that we might want to do is we might say, oh gosh, we have so much information here. There's so many different genes and so many different environmental factors. Can we narrow this down? Can we say, which genes or factors are sort of really associated with this health issue? And so we might want, for interpretability's sake, a very small subset of P. Let's say, P is this true number of covariates that we have. We want a really small subset of that to be able to talk about. And here we're imagining that P, especially as it arises in biology or medical examples, might be quite a bit larger than N being the number of data points. Okay, so what can we do? Well, there are a lot of options here. We actually could just go ahead and use the lasso. That would be a really sort of traditional thing to be doing in statistics, to use the lasso. But a problem with the lasso is that, at least as its vanilla framework proposes, it's an additive model. And so what's the problem with an additive model? Well, in particular, it's assuming something like the following. Suppose I find that there's some association between gene A and blood pressure, some moderate association. And I find that there's some association between the amount of sugar that you're eating and blood pressure, perhaps some moderate association. Now, that's the end of the story if I have an additive model. But I can imagine a world in which, maybe there's some association, perhaps moderate, between gene A and blood pressure, and some association between the amount of sugar you're consuming and blood pressure. But in fact, if you have gene A and you consume a lot of sugar, there's a huge association, maybe with your blood pressure level. And so in this case, we'd actually really like to be able to capture these kinds of interactions, not just have this additive model. And so that's essentially the question that we're asking today. How can we do this? We're certainly not the first to think about this. I'll talk about a lot of really cool work that's been done in this area, and that won't even cover it all. But a big challenge that all of this work faces is that now we have a p-squared dimensional problem. So if you think about trying to understand, every possible interaction and its association with blood pressure, well, if I have p covariates, now I have p-squared sort of questions that I'm trying to ask. And that's really the issue that we're going to be trying to address. That now that this is such a statistically and computationally hard problem, how can we get these pairwise interactions in high dimensions efficiently and in a meaningful way? Okay, so what we're going to provide is a fast and accurate method. And I think it will be no surprise, given the audience that we're talking to today, that it is a Bayesian method. And it's going to be for interaction discovery. And we'll see that it has better scaling in P and better accuracy than lasso-based methods. And we'll also see that it's orders of magnitude faster than naive approaches that you might take in Bayesian inference to this problem. Okay, so going forward, we're going to start by trying to set this up with a little bit more notation, this idea of discovering main and interaction effects. We're going to talk about our method. And in particular, if you're taking a Bayesian approach to a problem, basically you're going to have 3 steps, and we're going to work through those steps. So we're going to set up our prior and likelihood, our Bayesian generative model. We're going to set up how do we do fast inference. So, doing posterior inference, and then at the end of the day, you have to report your results. And you're typically not reporting the entire posterior, you're reporting some useful summary of the posterior. So we're going to talk about how to do that. And then finally, we'll see some experiments on both simulated and real data. Okay, so let's start with this setup. How do we discover these main and interaction effects? Okay, so we're talking about the exact same cartoon that we did before. We have all these covariates. Now let's add some notation. Let's call them x1 through xp. So remember, there are p total covariates. Let's say that we have a response, y. So here we have our response is just this blood pressure level. And so let's collect all the covariates into a covariate vector x. And anticipating that we're about to be doing a linear model, let's add a covariate that's basically going to represent an interceptor in that model. So that will just be this 1 here. Okay, so we have our linear model. Why would we want a linear model in this case? Well, when we introduce our parameter theta, theta tells us sort of exactly what we're interested in, the association between each covariate and the response. So the theta that corresponds to x1 is going to tell us the association between x1 and y. Okay, and the problem that we highlighted, sort of before we had notation for this before, was that actually, the problem here is that we have just this additivity, by which I mean you have this like theta 1 x1, theta 2 x2, theta 3 x3, and you're just adding them up. And there's no possibility for x1 and x2 to interact, for instance, for x2 and x3 to interact, and so on. And so there's a conceptually pretty simple way to solve this. Now we just introduce a new set of covariates, an expanded set of covariates. So let's call this phi2. So now we're going to have some new covariates, x1 times x2, x2 times x3, x3 times x4, et cetera. And we'll do this for every possible set of interactions. Pairwise interactions in particular is what we're looking at here, although I'll mention higher order interactions at the end. Okay, so that's great. Now we just expand our linear model to say, oh, it's phi2 of xn instead of just being, as before we had just the x's. Just this here. Okay, so now we have this expanded covariate set, and implicitly when I expanded this covariate set phi2, I've also expanded the parameter theta. So now theta is, technically speaking, it's not exactly p squared, but I'm just going to call it p squared because it's about p squared long as a vector, and we're interested in learning the elements of theta. Okay, so in particular, our goal here is really twofold, although I'm going to focus a lot on the selection aspect today. We want to select the non-negligible elements of theta. We're imagining that sort of, most of them are negligible. We want to select the non-negligible ones, which are the sort of covariates interactions that really matter, the main effects and interaction effects that really matter. And then we'd like to estimate those as well. We'd like to know what are those values in this case. Okay. So implicitly, and even just saying everything that I just did, I've made some assumptions, and I'd like to make those explicit now. So one of the assumptions is that this is sparse, that most main effects here are negligible. And so that's sort of an interpretability assumption, that I'm just interested in saying, oh, what are the ones that I can do some follow-up experiments on? What are the main effects that I can really use to summarize this when there are so many to begin with? Our second assumption is a more onerous assumption. This is a strong hierarchy assumption. And so this is a very common assumption. The idea here is that there is an interaction effect only if main effects are present. You could think of this as some kind of sparsity assumption on the interactions. So here we're saying, oh, if I have some moderate association between my gene and blood pressure and some moderate association between sugar consumption and blood pressure, then I'm allowed to have some association between the interaction of those two things and blood pressure. Now, I want to make a couple of points about this. So one, to the best of our knowledge, this is pretty commonplace in the literature. It certainly makes this a much more achievable problem. And our original paper that I'm talking about today does make this assumption. And all the experiments that I'll be talking about today make this assumption in some form. Although a paper that we just put on the archive that I'll only mention very briefly at the end is able to lose this assumption. And hopefully by going through everything that we do today, we'll kind of get a taste of how that could possibly happen. But essentially what we're doing here, the assumptions that we're making are sparsity assumptions, because that's what we want to get out of this problem. Okay, so we want to find this sparse collection of associations. And so why is this a challenging problem? I mean, gosh, this is just a linear model, right? Like, aren't we all super familiar with linear models? Well, the problem is that there are p-squared covariates. Again, I'm sort of eliding all the constants, but it's roughly p-squared covariates. There was an excellent session yesterday, how if you're trying to find a sparse set of covariates that are associated with the response, as a challenging statistical and computational problem, even when you are just trying to do that in, potentially an additive model or a model of this form that doesn't have all of these interactions. And so this just becomes an incredibly difficult problem when you now have all these interactions. You basically have p-squared to deal with instead of p. And so what we're going to do fundamentally, just at a very high level, is that we're going to use structure in the covariance. If these were any old covariates, like if they were x1 and x2 up to xp squared, you couldn't possibly get away from the p-squared cost. I mean, it would just be unavoidable. But these aren't any old covariates. Fundamentally, there's only p unique covariates. It's x1 up to xp, and then we're sort of multiplying them. Like they're like the terms you would get out of a quadratic. And so that's really what we're going to be using today. We're going to be using that special structure together with these sparsity assumptions to reduce to a problem that is linear in p. And that's really the key thing that we're providing. Okay, so now we have some notation. Hopefully that all seems straightforward. And what we're going to do is we're going to go ahead and say, what does our method look like? And in particular, again, thinking about the generative model, the inference, and the reporting. Okay, so why are we using Bayesian methods? Well, this will probably, again, be familiar to this audience, but of course, there are lots of awesome reasons to use Bayesian methods. We can incorporate expert information. There's certainly certain types of things that we want to get out of this problem that we can encode as being some sort of expert information. In this particular problem, we would be interested in uncertainty quantification. We're certainly interested in regularization. We really care about getting this, very small subset, the sparsity, and we care about the flexibility to encode what we think about, what we think is going on in the interactions. And so this all seems to really recommend itself to a Bayesian framework, that we get this nice graphical models framework. And so what is that going to look like? Well, again, there's these three steps. We choose our generative model, we compute our posterior, and we report the relevant summaries of the posterior. So let me just briefly overview these three steps, and then we'll go into more detail. So for the generative model, we're going to propose a new generative model. We call it the sparse kernel interaction model. And basically, in a graphical models framework, we'll encode sparsity and strong hierarchy. We don't see this step as being our principal contribution. We're really building on the shoulders of giants here. So Carballo et al. In 2009 introduced this awesome idea of a horseshoe that really allows sparsity and is encoded in the prior. Pirinen and Vitari in 2017 introduced a regularized horseshoe, which is really helpful in this particular case. Chipman in 1996 and Griffin and Brown in 2017 did amazing work on looking at interactions in Bayesian inference. And sort of the challenge at the end of all of this work is, how do we actually make this scalable to modern data sets with a lot of covariates in this interactions framework? And so that's really where we come in, is steps 2 and 3, making this something that we can do in linear time. So technically, our sparse kernel interaction model is new, it is different, but it's very similar to a lot of these existing models. Okay, so now what we're going to do is we're going to come in and we're going to provide a sampler that runs in linear time iteration. So I want to emphasize, on the face of it, there are p-squared parameters. And so how could we possibly get away from visiting every one of those in a sampler? That's what we're going to have to discover. We're going to have to say, hey, is there some representation that's only going to take time p to sample from instead of p-squared? And even if we did have such a representation, and you could do everything in time p, there's the issue of, okay, well, but at the end of the day, you have p-squared of these parameters. And so if you actually reported a posterior for every single one of them, that would take time p-squared. And so how can we get away from that? That's where we're going to have another kernel trick, our kernel interaction trick, to report all the non-negligible main and interaction effects in time p. And so you can already get a sense that this is going to have to rely on things like sparsity in order to work. Okay, so let's dig a little deeper into what exactly is going on in this kernel trick. And in order to do that, let's start thinking, like, how would you approach this problem if it were just handed to you? You know, you're talking to a scientist and they say, hey, I'm really interested in finding these interactions. What do you do? Okay. Oh, and I'll just mention before we go on that technically speaking, this sampler and this trick are not just for our particular model, the skim model, but the skim model is what we use in all of the experiments. And so that's what we'll see there. Okay, so what are we going to do? Well, I think a really amazing thing, and I'm like super sincere about this, a really amazing thing about Bayesian inference in the past few years is that we have these amazing automated ways to do inference now, with Markov chain Monte Carlo, with variational Bayes, with INLA, with whatever is of interest to you. And so let's consider doing that. How about I just say, hey, I have my generative model here. It encodes this sparsity and strong hierarchy. How about I just put it into my favorite, automated inference engine, like maybe Stan, which happens to be what we try here, and I just sample theta. So why is that going to be a problem? Well, remember, theta has p-squared parameters. So even just computing the likelihood for theta is going to take p-squared times n, and then of course I have to do that computation presumably at every step of a sampler, and so that's going to be problematic. And we see this. So I haven't told you what our method is yet, but let's start comparing it to stuff. And so here, what we're seeing in this particular example is on the horizontal axis, we're creating a bunch of simulated problems, and we're increasing their dimension, and it's on a logarithmic scale. On the vertical axis, we have the running time of the different methods. One of these is option 1, which I've just presented to you. Why don't we just put this into a fantastic sampler that we have. And then option 2 here is our method, what we're proposing to use, which we'll go over shortly. And we're seeing that we get this orders of magnitude difference, especially as p increases. The difference between p-squared and p is quite large. Now, for anybody who's familiar with Markov chain Monte Carlo, I think a very reasonable question that you're probably asking at this point is, okay, but what do you mean by running time? And so what we really mean is running 1,000 iterations of Stan. And so then there's, of course, the question of, did it mix? Did it actually mix? And we can check this. One way to check this is with this helpful heuristic, the r-hat heuristic. It turns out that if your r-hat for a particular parameter is greater than 1.05, then you might think that your sampler hasn't sufficiently mixed for that parameter. If it's less than 1.05, you might think that it is mixing reasonably well. And what we find, in fact, is that for every parameter being sampled, the r-hat is greater than 1.05 for this option 1, as I've just described it, and less than 1.05 for our method. And so in this case, it seems like, yes, this is 1,000 iterations, but there's more to it than that, that actually this is having quite a bit of trouble mixing, presumably because it's in such a high dimensional space in this case. And so whereas our method is not seemingly, at least according to the r-hat heuristic, having that trouble here. Okay, so what else can we do? Well, another option is to notice that, in fact, what we have is a linear model. In fact, the form of the prior is itself a Gaussian prior, and then it has some hyperpriors. And so we could just use conditional conjugacy. And again, when you think of, like, what are you told in your intro Bayesian class? Like, man, if you can use conjugacy, you're set, right? Everything is done. And so let's explore that idea. Okay, so if you've ever done anything with a linear model whatsoever, you know that you're probably going to, at some point, going to have to take something of the form covariate matrix transpose covariate matrix and compute it and invert it. And if you've ever done anything with a Bayesian conjugate linear model, you know that you're going to have to take something like covariate matrix transpose covariate matrix plus a Pyre precision matrix and compute that and invert it. Okay, so from the perspective of time cost, these are basically equivalent. So let's just focus on x transpose x. And then let's remember that our covariate matrix here isn't actually x, it's this expanded covariant space, this phi2. Okay, so when we have phi2, now what is going to be the cost of doing this? Well, we're going to have x, remember, let's recall that x is an n by p matrix. So phi2 is roughly n by p squared. And so let's think of our matrix algebra. We have an n by p squared matrix. We're pre-multiplying it by a p squared by n matrix. So we're left with a p squared by p squared matrix. And what does it cost to compute any of the elements of this matrix? Well, any particular element, you're taking sort of an n vector dot producted with an n vector, and so that's going to take time n to compute. Okay, so what does it cost to do conjugate Bayesian linear regression here? Well, to fill in this matrix, the matrix has size p squared by p squared, and the cost of any element is n, so the whole cost will be p to the 4 times n. And then inversion is somewhere between quadratic and cubic. Let's just say p to the 6th. It's all way, way, way more than p squared. So this, on the face of it, seems quite a bit worse than what we had on the previous slide. Okay, well, it turns out you can use some fancy matrix identities here and get this down, but it's still going to be p squared. You're still going to have that p squared cost. And we see this in practice. So again, haven't yet said exactly what we're doing, but let's start comparing it to these other methods. Again, on the horizontal axis, we have a bunch of simulated problems of increasing dimension. On the vertical axis, we have running time. We see that the naive approach in red is just going off the charts. Even with this Woodbury matrix identity, things are still pretty slow, and our method is quite a bit faster. And it's not just running time, it's also memory. So here we see that the memory cost is substantially higher by just sort of using the basic conjugate linear regression approach here rather than our method. Okay, so hopefully at this point, you're pretty excited to hear more about our method. So let's talk about what that is. Basically, we want to observe that kind of the problem here, I mean, the whole problem really, is this matrix, this phi2 transpose by phi2. It's too expensive. It's too expensive to compute. It's too expensive to invert. What are we going to do? Now, if you think about it, a type of conjugate linear regression that many of us at least are familiar with is basically Gaussian processes. We have this exact same problem in Gaussian processes, that sort of the underlying space is way too high dimensional. If you have a squared exponential kernel, it's infinite dimensional. And so what do we do to get around that in Gaussian processes? How do we avoid this really expensive computation and inversion? Well, one way to think about it is that we do prediction rather than posterior, getting the posterior just for theta. And so one way to think about it is that you're getting the posterior predicted, the theta transpose phi2 in this case, rather than just the posterior for theta. On the face of it, that sounds like a harder problem, like we're adding this extra phi2, but let's see why it's actually easier computationally, at least get a little bit of intuition. So if you think about it, what's happening here is we're computing a posterior uncertainty. We're computing a posterior covariance matrix. And so this is a posterior covariance matrix for theta. And so if we're multiplying theta by phi2, if we want to change that posterior covariance matrix, we're sort of pre and post multiplying by phi2. Okay, so we still have this phi2 transpose phi2 buried in here, but the key is that actually we can just compute phi2 transpose, multiply it by itself, and that'll turn out to be pretty fast. So somehow it looks like we're doing more, but we're actually having to do less. Okay, so let's check that this actually is going to be less expensive. So let's focus on this phi2 transpose. What's going on there? Okay, well, we now have, as before, an n by p squared matrix. The only difference is now that we're post multiplying by p squared n matrix. So pretty similar setup. The result is that we get an n by n matrix. And remember, we're assuming that p is much greater than n here. And so, you know, if anything, this cartoon is not sufficiently exaggerating how big p squared is. What does it cost to compute one of the entries of this matrix? On the face of it, if we didn't think about it too hard, it looks like it's going to be a p squared vector dot producted with a p squared vector. So that's going to cost p squared. But this is where our kernel trick comes in, or any kernel trick comes in. The observation is that this isn't just any p squared covariance that we're dot producting with a p squared matrix, or sorry, with a p squared vector. These are these extremely structured p squared covariance. This is like saying if I took the quantity parenthesis x1 plus x2 plus x3 up to xp n parenthesis squared, yeah, that has, you know, if you wrote it all out with algebra, that's going to have p squared terms in it. But you could just compute it in time p and then square it, and then it only takes time p. And so that's exactly the idea that we're using here. It's a little bit more complicated, but really it's fundamentally that idea. And it's fundamentally something like a polynomial kernel that we can use. And so instead of this taking time p squared, it's going to take time p, precisely because of a kernel trick. Okay, so then the observation is, what is the total time cost? Well, we compute the matrix. It's got n entries by n entries, so that's n squared. And the computation of any entry is p, so we get pn squared. And then the inversion, again, is between quadratic and cubic. Let's call it n cubed. The point is it doesn't have any p dependence. And so we're left with something that is purely linear in p. Now, of course, the elephant in the room is that we still have this like n cubed dependence, and that's not great either. We'd like to get to high n as well. But if p is what we're really concerned about, and certainly in these biology and medical applications, that is often the case. If p is very large, this has just totally changed the game. We don't have this p squared dependence anymore. We just have p. And I'll talk a little bit about the n thing at the end of the talk. Okay, so we can see now why this would be so much faster in those examples that we saw earlier, why it would be so much faster than sort of very reasonable things that you might have thought to use to do sampling, to do a conjugate approach to this. And so now let's take a little time to compare this to lasso-based methods, because there are lasso-based methods that you can use in this for interactions. Okay. Now, before we get to that, there's technically an issue, and that is the following. Yes, we have the sort of posterior of theta transpose phi 2, but actually what we're trying to get is the posterior of theta. We want to report which theta values we are confident are non-zero or non-negligible, and which ones we think are just really not meaningful. And so on the face of it, it seems like we're gonna have to do a little bit more work. We don't just get those for free out of this equation. We're gonna have to say, how are we going to get those theta values, and make sure that it doesn't take like p squared time. Okay, so our goal isn't just to do prediction, which is what often happens in Gaussian processes, so you're all done here and then you can just sort of, step away. Our goal is to find these main and interaction effects, so we have to do a little bit more. Okay, so the first step that we're going to do is we're going to report these main and interaction effects in constant time. So what I mean by theta sub xi is the theta that corresponds to the xi term. What I mean by theta sub xi xj is the theta element that corresponds to the xi xj term. So basically the main and interaction effects. Okay, how are we going to do this? Well, let's let EI be the ith standard basis vector. And now we have some general theory, but honestly, it's probably just a bit hard to read directly. I think an example is going to completely clarify how this is done. Let's look at G, this Gaussian process, up top, applied to this ith basis vector. Okay, so if I take the ith basis vector and I put it into phi 2, that means I'm going to get zeros out everywhere in phi 2 except for the xi term, and I'm going to get 1 squared at the xi squared term, and everything else is 0. And so when I multiply that by theta, I'm going to get out theta sub xi plus theta sub xi squared. And I can do the same thing with negative of the standard basis vector. Then I'm going to get zeros everywhere in phi 2 except for a -1 at the xi term and a -1 squared, or 1 at the xi squared term. Now I can just add these together and I'll get theta sub xi. And everything is very convenient because everything's Gaussian, and so you can really just get the whole distribution here. You have this nice Gaussian distribution. Okay, so this shows us that we can get the posterior distributions for a main effector and interaction term in constant time once we have this full posterior. But that's not enough, because there's still p squared of them. And so if we reported every single one of them, it would take time p squared. And so finally, we're going to use the sparsity assumptions. First, we assume that k much less than p, by the sparsity assumption on the main effects, that k is much less than p. The number of main effects that are non-negligible is k here. That we can get that in OP time by just going through every single one of the main effects and reporting it. So just go through all p of them. But now by the strong hierarchy assumption, we only have to look at the k squared that were associated with those sparse main effects, and we can report them. Now, something that hints at how we could get away from this is you'll notice that somehow we're only making this assumption actually in the reporting rather than the model itself. And so if we could find a way to really have this linear representation of what is actually contributing to any effect and learn with it do sampling with it and report with it, then we can get away from this. So that's just sort of a hint of how we might get beyond this strong hierarchy assumption, but certainly we did make that assumption in this work. Okay, so finally, let's look at some experiments. Now, here's an interesting observation before we get into the experiments, is that this is a tough thing to evaluate. Like, evaluation also came up in Suchi Suriya's talk. Something that is pretty straightforward in some ways is something like you're doing classification or regression, and you're looking at classification error or regression error. At the very least, there's some widely accepted ways to evaluate there, and it's easy to do, but it's not what we actually care about here. We care about recovering whether covariates are associated with some response in a meaningful way that is sparse. And so that's tough because we don't have ground truth in any real data set. We don't know what is truly associated with the response. And so we're going to look through some possible ways to get around this, but I just want to put it out there, first of all, that this is a challenging problem to say how well we're doing. Okay. So before we get into that, an easy thing to do is just time things. Like let's do timing, see how we do on timing. And also check that we actually are competitive with lasso-based methods, because even if these awesome Bayesian methods are like super more accurate and better, I've definitely encountered, talking to practitioners who are running their lasso-based methods. They're like, well, yeah, but our lasso method takes like a second, and so I'm not gonna do something else. And so let's make sure that we have something competitive here. Okay, so for lasso methods, both for timing and quickly we're going to get to performance as well for accuracy, we're going to compare two lasso methods. So one, we're going to call pairs lasso, and this is just applying your basic vanilla lasso, but to the expanded covariance space. Two, there's a method called hierarchical lasso that was specifically developed, e.g. in this Lemon-Hasty paper from 2015, to deal with this association problem, this interactions problem. And they both take time p-squared perturbation. So we expect that if you get to sufficiently high p, that our method should be doing better because it just has this p dependence. But of course, we're using Markov-Chapman-Carlo. That could be quite slow, and so we want to check that it's not too crazy for the earlier p's. And so that's what we're seeing here. So on the horizontal axis, we're seeing the dimension increasing, again, on a logarithmically growing scale. On the vertical axis, we have running time again. It is the case that our method is slower in the beginning, but as expected, when we get to a sufficiently high p, it overtakes these other methods. Okay, so it sounds like this is something we can run, that we can even compare, that people might be willing to use. And so let's go ahead and check out performance. How does this look in performance? Now, as I said, there's this issue of what is ground truth. And so the easiest way to get around that is to just simulate data sets. We're going to talk about some issues with that and why we want to get away from purely simulated data in a moment, but let's just start off with simulated data. So we're going to do 36 different simulated data sets. This means that we know the ground truth effects. We know the thetas because we made them up ourselves. We're going to take p up to 500, so up to 500 covariates. That means that there are 125,000 total parameters. That is, I struggle to believe that anybody would not call 125,000 parameters a high dimensional problem. That is a very challenging problem. Now, if we had 500 dimensional problem, that would be a lot more palatable. That would be a lot more like something that we can handle. And so that's the idea, is that we're trying to not have a problem that's truly 125,000 dimensional. We're trying to really have a problem that's just 500 dimensional. Now, in our paper, we look at both estimation and selection. For the purposes of our time together in this talk, I'm going to focus on selection, but I'll encourage you to check out the paper for estimation. And what we're interested in here is getting as many possible correct effects and as few possible incorrect effects. So correct effects are things that are truly effects. We know which ones are the non-negligible effects here. And incorrect effects are things that in the simulation were totally zero. They didn't have any actual association, but an algorithm incorrectly picked them up. Okay, so I'm going to show a plot, and then I'm going to describe everything that's going on in that plot, because there's quite a bit. Okay, on the horizontal axis, we have number of correct effects. On the vertical axis, we have number of incorrect effects. Let's go ahead and look at this little red plus that's here in the upper right-hand corner. That is an instance of the hierarchical lasso applied to one data set. So here we have H lasso, this is a hierarchical lasso. And so what's happening here is that this particular data set, on this particular data set, the hierarchical lasso is picking up all 5 correct effects, because there were truly 5 correct effects, and for the main effects. So this is just looking at the main effects for the moment. And it's picking up over 60 incorrect effects. So that's sort of over an order of magnitude more incorrect effects than correct effects. And that's problematic because, you can imagine the hope here is that somebody's going to come in and do some cool scientific experiments after finding these associations. And it seems like they're going to waste a lot of time on things that we might hope that we would not have picked up in the first place. Okay, so when we look at all the hierarchical lassos applied to different data sets, we're seeing that they're picking up lots of correct effects, but just tons of incorrect effects. When we look at pairs lasso, so pairs lasso is here in blue, that's these little crosses, it's picking up fewer incorrect effects, but very few correct effects. And of course, the whole point is we want to pick up as many correct effects as possible too. We really want to make sure that we're getting out any interesting thing that's going on in this data set. So that's not great either. What we really want is to get a lot of correct effects and very few incorrect effects. And so now here in these green dots, we're seeing our method applied to all the same data sets, and it's picking up all the correct effects, or at least many of the correct effects, but very few incorrect effects. And we see this pattern as well for the pairwise effects. This is the same sort of plot, number of correct effects and number of incorrect effects for the pairwise effects. We're seeing that hierarchical lasso and pairs lasso is picking up lots of correct effects, but also lots and lots of incorrect effects, way more incorrect effects than correct effects. And our method is picking up very few incorrect effects, but seemingly at least as many correct effects. So that looks pretty good. But I think very reasonably, you might say to yourself, yeah, but simulated data isn't like real data. A really well-known issue with simulated data is that the covariates are typically, is the easiest way to make simulated data, and certainly what's happening here, is the covariates are independent. And real data does not have independent covariates. And so what could we do to have more realistic covariates? Well, one step further along is that we could take real life covariates and just simulate the effects. So we take all of our covariates from a totally real data set and simulate the effects. Here we'll simulate 5 true main effects and 10 true interaction effects. The covariates are from this residential building data set. This is a publicly available data set. And like a lot of real life covariates, they're highly correlated. So 20 out of 105 of them capture 99% of the variance here. Okay, so what we're going to do is we're going to do the same check that we did before. We're going to say for each method, how many correct effects does it pick up? As many as possible out of these 5 main and 10 interactions would be ideal. And then how many incorrect effects does it pick up? How much time are we going to waste on getting all of these incorrect effects? Okay, so first let's look at pairs lasso. So what we're seeing here is that first, in terms of main effects, this method is picking up 2 correct effects out of a total of 5, and 5 incorrect effects, so more incorrect effects than correct effects. Out of the pairwise interactions, it's picking out 3 of the correct effects out of a total of 10, and 21 incorrect effects. It is not able to, not get rid of, it has this sort of order of magnitude more incorrect effects than correct effects. Okay, so the hierarchical lasso can also use this. It's picking up a little bit more in terms of correct effects. It does seem like, roughly this many correct effects is what it's picking up. But it's picking up tons of incorrect effects. Both main and pairwise effects, we're getting 19 incorrect effects from the main effects and 18 incorrect effects for the pairwise effects. Okay, and then how about our method? We're able to pick up just as many correct effects as the other method, so that's good. Sort of not losing out on anything that you wouldn't have lost out on in these cases. But crucially, we're picking up absolutely 0 of these incorrect effects. And so it seems like you'd be wasting less time on all these extra incorrect effects. If you were doing some sort of follow-up study on this, you're not picking those up. Okay, but again, you might be saying to yourself, yeah, but well, in real life, maybe it's not a perfect linear relationship between your covariates and your response. How do we capture these real life relationships? How do we know that this is going to work in a truly real data set? Okay, so let's try to go a little bit farther in that direction. Let's look at something that is mostly real data. Okay, so, well, let me explain what we're doing. So we're going to take both covariates and response from a real data set, but by design, it's going to be a super low dimensional data set. So in particular, we're going to have P equal to just 6. Why are we interested in getting a low dimensional data set, given everything we've just said about being really interested in high dimensional P? Well, we're going to augment this with some totally fake covariates. And so the key here is that since most everything is fake, we're going to get a lot of information about what's picking up incorrect effects and not. We don't know what's going on in the real data set, so it's nice that it's small because it's kind of isolated, but we do know that everything else, these 200 dependent covariates, are just totally fake. And so we know if anything picks them up, that this is going to be a problem. And so in the end, we have over 21,000 total parameters here. This is a very high dimensional challenging data set. Wouldn't it be nicer to work with effectively 200 parameters instead of 21,000? Okay, so this time the key is a little bit different. In the last case and the case before, we simulated the effects. So we knew exactly what were the true effects. And so we could say higher is better for that. In this case, we are not simulating the effects. So there is no order to the original effects. It would be nice if all of the methods picked up some non-zero number of effects. It's sort of disappointing if we don't get anything from the data set. But otherwise, there's no order here. We don't know that higher is better because we don't know what is the ground truth. We do know that anything of the fake effects, any of these totally fake effects that we just added arbitrarily, are wrong. And so there we definitely know lower is better. We don't want to pick up any of these totally fake effects. Okay, so let's look at these methods again. So we'll start with pairs lasso. So here, you can look at the main effects. It's picking up 4 effects from the original data set. Again, it's nice that this is non-zero, otherwise we can't really say too much about this number. It's picking up 0 fake main effects. That's great. That's the kind of performance that we want. It's picking up 2 of the original data set pairwise effects. That's, again, it's nice that it's non-zero, but otherwise we don't have too much we can say about this. But it's picking up 78 fake pairwise effects. That's humongous. That's absolutely dwarfing the number of possibly even real main and pairwise effects that could exist. Okay, with hierarchical lasso, we're having a similar set of issues. It's picking up a non-zero number of original effects. That's good. Otherwise, there's no ordering here. We can't tell that hierarchical lasso is better than pairs lasso or otherwise for the original effects because we don't know which ones are truly there. But we see that it's picking up tons of fake effects, these 46 fake main effects and 38 fake pairwise effects. Again, that's totally dwarfing the number of even possible original effects that we could have had. Okay, and then finally, you can run our method. It's picking up 3 main effects and 1 pairwise effect from the original data set. Again, it's nice that that's non-zero. Otherwise, there's no ordering here because we don't know the original effects. We don't know which ones are actually there. But it's picking up absolutely 0 fake effects, these totally random appended fake effects. And so if anything, that feels like I probably can trust the original effects that it's picking up more because we see that it's just not picking up these fake effects that have nothing to do with the data set. Okay, so let me go ahead and make some concluding remarks. So we provide here, what we focused on here today, are these fast and accurate detection of pairwise interactions. Our paper appeared in ICML in 2019, the kernel interaction trick, fast Bayesian discovery of pairwise interactions in high dimensions. If you're interested in this, and I hope you are, I encourage you to check out the archive version because it has some corrections relative to the ICML version. So definitely check that out. We have some code in that original paper, and you can find it linked from that paper. But some amazing folks, not us, just generally amazing folks, have added this to Prado. So as I said before, I think a really amazing thing now is the automatedness, the ease of use of these modern probabilistic programming languages. And I think this is really great that this has been added to Pyro, so I want to just point that out. Today, I really focused on pairwise interactions, and certainly that is what all of our experiments focus on, and even in our new paper, which I'll mention very briefly shortly, we focused on, but everything absolutely goes through for higher order interactions. Just like if you take x1 plus x2 plus x3 up to xp and cube it, you're going to get a cubic number of terms, but you could compute that in linear time, that quantity. It's the same thing here. And so you can absolutely do higher order interactions. And the only reason that we sort of don't have that in our experiments yet, at least that we've done, is that we haven't yet been, I mean, we're starting to have some talks with biologists now that might change this. But so far, we haven't really seen a case where somebody really cares about higher order interactions. Certainly, we've seen a lot of cases with pairwise interactions. This seems to come up a lot in biology, at least in our discussions with biologists. But the higher order one we hadn't yet seen, although, and so I want to say if anybody has a higher order reaction that they really care about, I'd love to chat. Okay, so what are we doing next? Well, one big issue that you may have noticed here is that this all makes this linearity assumption. So that I have this linear model. I'm assuming that my covariates are associated with the response via this linear model. But of course, tons of real life things are not linear. They might be, quadratic, they might be sinusoidal. You can imagine all kinds of potentially nonlinear relationships that very naturally arise. And so what we'd really like to be able to say is something like, oh, I have some function that relates my covariates to my response. Can I express it purely as functions individually of each covariate? Or do I need to have interactions? Can I make this a sparse expression? Can I express this relationship only as functions of a very small subset of the covariates and a very small subset of potentially functions that involve just two of the covariates and so on? And so this is one of the questions that we are answering in a paper that we just put on the archive. I mean, this is hot off the presses. The SCIM-FA kernel, high dimensional variable selection and nonlinear interaction discovery in linear time. So I'll encourage you to check that out there too. It also links to code, and we're trying to put, Raj is putting together this really nice tutorial. So hopefully that'll be up soonish. It's not up just yet, but it should be soonish. Also, Raj is giving a talk about this at ISBA 2021. And by is giving, I mean gave on Monday, but everything is online now. And so you can totally check out that talk if you haven't already. And I'm sure he would just love to chat about this stuff too. Another really big thing in this new piece of work is removing this strong hierarchy assumption. And again, I want to just give this at least slight intuition that what we were doing in the paper that I talked about today was we were really enforcing the strong hierarchy assumption at the reporting level rather than the model level. So at least that gives some hint about how we might be able to get rid of it, is if we could really focus on the model level and a better way to do reporting. And so that is certainly something that we look at in this new paper. An elephant in the room here, as I mentioned before, is the scaling at n. Certainly there are many cases where we really want a large P. We can imagine lots of biology cases where you can easily get just hundreds of n, but thousands or millions in your P size. And there are yet still cases where n can get much larger than the hundreds. And of course, here we've only seen n in the hundreds today. And so of course we want to improve the scaling in n. Now, in some cases, in some sense, this is an old problem. I mean, this is the problem of Gaussian processes. I think there have been amazing talks, even at this ISBA, and certainly at other ISBAs, about scaling Gaussian processes better in n, because they make exactly this trade-off. I will briefly mention some work that we've done in this area. So we have this nice paper at AIStats 2019 that focuses on, we often are interested in theoretical guarantees when we're improving the scaling at n. We're really focusing on the things that we are often reporting in Bayesian inference, like a posterior mean, a posterior variance. And so how can we scale Gaussian processes and get guarantees on those quantities that we report at the end of the day? So that's this first paper here from AIStats 2019. Also, we've done other work in scaling basically the kernel matrices that go into these things, like this data-dependent compression of random features that uses some of our core sets work, if some of you are familiar with that work. And of course, there's tons of other great work on this. One thing I will say is that this isn't immediate, at least like some thought has to go into this, because a lot of the work where we're looking at this scaling, a lot of that work is based on Gaussian processes for spatiotemporal models, right? So one of the big reasons that we use Gaussian processes, I think a huge success story of Gaussian processes, is doing some kind of regression in time or space. But then what often methods are taking advantage of is localization. That somehow, as you go far away from a particular point in time or space, that you're losing a lot of that association. And so we have to be careful that we're doing something that works for polynomial kernels here, because that has a very different flavor than those spatiotemporal kernels, like squared exponential. You almost have the exact opposite sort of issue of localization with a polynomial kernel. And so we'll have to think about that for scaling in n. We're giving it some thought, but we'd love to chat with anybody who wants to chat about these things too. And a big thing that we're focusing on now is applications. So we're starting to talk to some biologists, and we're starting to talk to some people in the medical field, but we would love to talk to anybody who is interested in this. Certainly, we are not the experts in those fields, but we're hoping that this could be useful in those fields, and it sounds like it may well be. I'll just take this question very quickly. So on slide 14, did we look at the results without including the fake noisy covariates? I don't remember off the top of my head what they would be without, but I mean, it's a pretty straightforward problem without. So you can just say, oh, you know, I'm going to do this regression, and since it's so low dimensional, you expect that it should work out pretty easily. And so I would expect that we would have, you know, some pretty sound things that we would agree with. One thing I will add, though, just because this was asked, and I think it's a fun point, is something that we have looked at in the new paper. Because something you could say is, so we had added all these fake noise covariates, and with the fake noise covariates, they were these independent covariates again, right? And we had just said, oh, independent covariates, like real life isn't like that. And so something that's cool in the new paper is that we append a bunch of fake covariates, but from a different data set. And so they aren't totally independent, they actually are related to each other. And we find a similar set of results in this case, that we were able to avoid the fake noise covariates, but other methods seem to have trouble with that. Since I think I have a little bit more time, because I think 3 is my hard cutoff, I'm just going to use this time to briefly point out some other really fantastic work by people in my group and collaborators that we've been working on that you can see at ISBA, and I'll sort of plug that a little bit. Also, I'll just note that if anybody is looking for a postdoc or a PhD position, or something else, maybe you're interested in some other kind of relationship, I would be very happy to chat about that. Just come and you know, give me a ping in some form, maybe the Huba app, maybe on email, something else. But we certainly have open positions. A lot of people are graduating, which is awesome, and starting faculty positions. And so, so we have some open positions. And so with that, let me just also talk about some of these awesome people and the things they're doing. So as I mentioned, Raj's talk was on Monday, but you can still see it and you can still chat with him and ask questions. And that's all still available. So I'll, you know, certainly encourage that. Nick Bonaker is giving a talk on using Bayesian inference to help people with severe motor impairments. So these are people beyond paraplegic, beyond quadriplegic, can use Bayesian inference to help them type and draw and use GUIs in the way that we're all used to. Diana Kai is giving a very fun talk about some dangers of using priors with Bayesian inference for the number of components and how things might go wrong. So I'll encourage you to check that out tomorrow. Soumya Ghosh, our excellent collaborator, just gave a talk on approximate cross-validation and how to speed up, you know, cross-validation via approximations for structured models. Ryan Giordano has a super cool talk. He's been doing a lot of work on the infinitesimal jackknife. He'll be talking about how we can get frequentist variability of Bayesian posterior expectations in a really fast and nice way. Lorenzo Masuero is talking about how to know how much more we might learn from data if we got more data, especially in sort of genomic studies. Tin Nguyen is talking about, in Bayesian nonparametrics, we're often using some kind of finite approximation. Can we talk about the quality of different finite approximations and how they compare? Brian Tripp is talking about how sometimes risk isn't always the best way to compare estimators. Can we do it better for a particular data set with C-values and decide whether to you know, choose a particular Bayesian estimator that way? And Will Stevenson is talking about measuring the sensitivity of Gaussian processes to the kernel choice. In our particular thing that I just talked about here, our kernel is kind of set. You know, we know the kernel that we're using, it's this polynomial kernel, but in many cases, you actually have a choice that's expressing prior information, and you might have some questions about it. Okay, thanks for bearing with me there. And I'd be happy to take any more questions after, I believe, first we're going to have the discussants, and then I'll do the rest of that. Thanks.
Thank you, Tamara, for a wonderful talk. So we move to our first discussant now, Sylvia Richardson.
Thank you. Let me just try to make this full screen. There we are. And I wanted to try to be fancy and have the pointer. It's spotlight. There we go. So thank you very much for inviting me to discuss this important paper, and congratulations to Tamara and co-authors. So I will just structure my remarks. Yes. And I will report on some numerical experiments which highlight some different aspects. I will give an example on pharmacogenomics, where interactions are really important, and just finally a few comments about the detection of higher order interaction. But first, I want to acknowledge that this discussion has prompted a group effort between a group in the BSU and a group at the University of Oslo, because we are part of an EU project on breast cancer and finding a new treatment, by using combination of drugs in breast cancer. So there's really strong interest in interactions. So I want to thank them very much. So I will use very much exactly the same notation, the same framework. So we're in this linear framework with basically an expanded vector of covariates. Now, Tamara already talked about comparison to the hierarchical lasso, which here is implemented through, which uses strong hierarchy as well as all the other tricks of the lasso to be feasible for moderate P still. Another algorithm I wanted to mention, which we've done some comparison, is the XYZ algorithm of Thanai and colleagues. This is different in the way that it does not make the strong hierarchy assumption. It's still sub-quadratic in P, and it managed that by using a combination of random projections and efficient sorting algorithm to actually target the interaction. So I'll show some comparison with this algorithm. So SCIM basically is a Bayesian, fully Bayesian algorithm, and besides the strong hierarchy, the sparsity is implemented. I mean, I don't think Tamara had time to go into the detail, but if you go down in the very nice model in the detail, there's this horseshoe prior. And among the horseshoe prior, there's a certain number of parameter which kind of controls. In particular, here I would highlight S, the expected number of significant main effect, and show how this parameter is influential. We haven't studied all the others. Now, with a very nice kernel trick, this can be made indeed and it's fantastic in a problem of linear, in P. Nevertheless, the posteriors need to be recovered from the GP, and there's these post-processing steps. And we had found that the post-processing step does incur additional costs. So now just go through some of the numerical experiment to highlight some of this, but this is what Tamara had shown, and clearly indeed you could see that the slope of the hierarchical lasso is steeper than that of SCIM, because as we know, SCIM is linear in P and not the hierarchical lasso. But nevertheless, the overall computational efficiency will also depend, as Tamara mentioned, of a combination of P and N. So this is, again, on computational time, results of some experiment we've done. Here, N equal 100, when then the plot before N was 50. And on the left-hand side, we've looked at basically the increased computational time of the post-processing. We've also run way behind 500, going up to 2,000 covariates and the interactions. And you could see that the actual as it's implemented, the post-processing cost becomes prohibitive. Here on the right-hand side, you see that XYZ is extremely computationally efficient, and that the Pyro implementation, which is not fully based on, because it's based on a map estimation, is on par with the hierarchical lasso. Now, we also wanted to look at the performance in detail at the performance in terms of true positive, false positive. So again, as Tamara said, synthetic data set, very similar setup to what was chosen in the paper, except we have 100 observation there, and we go up to 2,000 covariate. We also look at the effect of not meeting the strong hierarchy assumption and having the strong hierarchy only partially met and not fully met. So as absolutely illustrated by Tamara, so here I'm only reporting SCIM, the Pyro application of SCIM, not the fully based because it wasn't feasible. You could see that it's absolutely brilliant in terms of controlling the false positive, whether both the hierarchical lasso as well as XYZ, which is in purple, have a number of false positive. But if you look at the true positive, there's, if you look at the trend, so the SCIM, there's two versions of SCIM here with different number of this S, which was the expected number of main effect. And here we've used 3 times the number of simulated one. And here we've used the exact number of simulated one because it's a simulation. And you could see that when P gets larger, then basically it becomes, SCIM becomes very conservative. Either version of SCIM become very conservative, but actually that having S larger, because it would encourage less conservativeness, does better. If you look at the impact of not having the fully meeting the hierarchical assumption, you could see that it does impact it does impact SCIM quite considerably. Here we were 80% for a small p, and now we're down to roughly 50%, where it doesn't impact either the hierarchical lasso or XYZ. We see more or less the same curves. So basically, in terms of the numerical conclusion, it absolutely no false positive, but of concern, the sensitivity can be quite low, and also it will suffer when the strong hierarchy is not met. I think there is a lot more understanding to do about some of the hyperparameter prior choice. We've just done one comparison, and I think it'd be really interesting beside the default choice to actually dig more into, the horseshoe default priors to actually improve the performance and maybe the sensitivity of the method. I now want to go on to the, this pharmacogenetic application, which concerned melanoma. So basically, Tamara wanted a biological application, a biomedical application. So here is one domain where interaction absolutely crucial, because basically by doing many testing in vitro against a library of drugs and combination of drugs, which people are trying to find new treatments or more efficiency treatment. So we were lucky to have collaborated through our Norwegian collaborator to have access to a melanoma data set of drug efficacy, which is absolutely huge because there's many drugs, many cell line, and so on. How do you measure drug efficacy? You just see whether the cancer cells are killed. So 100% is brilliant, you've killed all the cancer cells, 0% means the drugs have done nothing. We've only analyzed a subset of 500 of these experiments, and the covariates are either the mutation of cancer-related genes, so occurrence, yes or no of mutation, continuous gene expression, and also the pathway, the certain pathway that these that these drugs were targeting. And we're interested in looking for either drug-drug interaction or drug-cell line interaction, because what you want often is that a mutation, a specific mutation, will mean that the effect of the drugs can be enhanced. So applying SCIM, the full Bayesian SCIM, to this subset of 500 experiments, this is what we get for the main effect. I mean, it took 11 hours, so it's quite a challenge, to run it, but we get, as expected, quite a lot of all the main effects of the drugs and a couple of other main effects concerning the mutation. But we have lots of prior information, but we had to lower basically the cutoff, because if we were using the default cutoff, we would get nothing, basically. Nothing would have come out. But we know these drugs are doing something. Of course, there's a lot of biological variability. So here, lowering the cutoff, we take 12 main effect forward, and we find two very interesting and known, completely making sense interaction. One, a drug times cell line interaction in the BRAF pathway, and another one, which is a drug-drug interaction. So doing this cutoff, a much more lenient cutoff, we find indeed two interaction which are totally believable. But what I'm moving towards my last point is that sometimes what you are interested on top of that is the fact that drug-drug interaction can hinge on specific mutation, i.e. whether you have a combination of drug and a particular mutation, then you could have more efficacy. So we're now trying to look at interaction of order 3. And as you can see here, this is definitely evidence of efficacy, which is much increased when you've got a mutation in the BRAF pathway, which is one of the main pathways involved in melanoma. So we just thought, well, great, we've got a way forward given in the paper to actually extend, the method for looking at three-way interaction kernel. And so we tried to extend SCIM in two ways, either by extending the specific two-way SCIM kernel and doing exactly the same methodology, nice to the power 3 and so on, or starting from the general R-way kernel, which is in the appendix. But in trying in both ways, we hit some obstacles. When we did it by extending the SCIM kernel, some terms do not cancel out. And then basically, if you take that way, you've got spurious constraints that are inappropriate. And if you try to go through the general route, then we did not find solution to the set of equation either. So our question to the author is, we feel there's still a gap to be solved to generalize, as in the paper, beyond two-way, and it would be great to have a working example for the three-way interaction, because we are very interested in three-way interaction. So in summary, it's a very elegant approach. I think there is, but as Tamara said, there's still this bottleneck in N, which may limit applicability, but that's not the main point of the paper. SCIM, as it is implemented or recommended, is highly conservative. And I think better understanding the influence of some hyperparameter would improve the performance. There are cases, completely biologically meaning, where strong hierarchy is not plausible, and I've shown one. We think that maybe XYZ, which has quite higher false discovery rate, but could possibly be improved by better tuning. We're very interested in extension to higher order interaction, but we don't believe it's fully resolved from the paper. Thank you very much, and congratulations.
Thank you, Sylvia. So our next discussion now, David.
Share my screen. Okay, can you see it okay? Yeah, awesome. So yeah, in my role as the second discussant, I thought not having access to Sylvia's discussion, I wouldn't comment in detail on Tamara's approach, because then it would become repetitive and boring. And so we'll see what I do instead. Okay, so my opening comments, I'd first like to give Tamara a really big congratulations for a well-deserved award. I think, like, over the years, it's been really delightful to know her and see her at conferences, and also to read a lot of her really interesting work on core sets and other ideas. I've really constantly looking at her papers and finding interesting ideas. And I think that this is no exception. I really enjoyed this idea about computational scalability and interaction modeling, which is really quite clever trick, I think, and quite useful. I think that some of the things that Sylvia saw were like just a symptom of doing high dimensional Bayesian interaction modeling. Like you put in the horseshoe, you have really really large P, like something you have to give, either you incorporate the Bayesian machinery includes a multiplicity adjustment, in which case you're controlling like the false positives, but the power is going to go down a lot, or you have a lot of false positives like the other methods have. So it's really hard to find interactions. You need to have really, really high signal to noise, which we often don't have in biomedical studies. Amy Herring has a student, Fook, who's doing some simulation studies motivated by our interest in our environmental health. And it's finding a lot of these methods have really, really low power for the types of sample sizes that we see in practice. And so that it's just a hard problem. If you're just trying to tell from the data whether there's an interaction, particularly a three-way interaction, but two ways is bad enough, then you're going to miss a lot. You're going to get either a lot of false positives or a lot of false negatives, just because there's not enough information in the data, particularly when you have correlated predictors. Okay, but before going on, I'd like to also mention that Susie Bayarri, she was around Duke a lot, so working with Jim Berger, so I knew her really well, and she was really amazing. So I'd like to kind of mention that. So she had an amazing intellect. She had really cool work that she did. I spent a lot of time talking to her. She was also like an amazing personality. She was very kind, curious, adventurous, kind of always in a good mood, energetic, interested in talking about ideas. And so I really miss Susie myself. And I think that it's really fantastic that ISBA is honoring her legacy and also a young talent like Tamara with this award. So this is great. Okay, some comments on interaction detection. So this is a timely and important area. It arises in many application areas. I'm commonly motivated in particular by environmental health applications. I'm PI on an R01 right now, funded by the National Institute of Environmental Health Sciences, which is focused just on this problem in environmental health. And so we're constantly having problems like this. And so we have some sort of YI, which is some health outcome or outcomes for individual I in an epidemiology study. And we have some sort of vector of exposures, X1 to XP. These could be things like air pollution in air, but more commonly in the studies we're looking at, you might get something like a blood sample and measure a bunch of crud in somebody's blood in the blood sample, different heavy metals and phthalates and bisphenol B and all this stuff. Yeah, so you might get this kind of very long list of things that you're exposed to. We would also have covariates as well that we want to adjust for, like ZIs, but I won't talk about that today. Okay, and so this is what's known as the mixture problem. And so there's a lot of interest in this. And so we might be exposed to a mixture of different exposures. And so classically in toxicology and epidemiology, you look at one exposure at a time, and that's sort of well developed. But what happens if you're exposed to a mixture of correlated exposures? And so this can happen even through like the thing you're exposed to contains a bunch of constituents, and disentangling the health effects of those different constituents is a really difficult problem that we've been interested in. A big focus of NIHS as well. So some challenges in this mixtures of exposures setting. The exposures can be high dimensional. Sometimes they're not like uber high dimensional, but even in moderately high dimensions, interaction detection is very difficult. And so if you have dozens or hundreds, that's really quite huge in this setting. And some of the X's can be quite highly correlated as well. And so you also have some issues, have some sort of annoying issues, like if the X's are low, within some sort of standard deviation of the assay being used, they don't like to report the actual value, and so they might give you some just below the limit of detection, so you get this kind of censoring. The dose response curves are typically nonlinear. I think that in Tamara's defense with using linear methods, as long as it's kind of monotone or roughly monotone, you can often pick up if you're just thinking from a testing perspective, but the curves are often kind of nonlinear. They might go up, and then there might be like a downturn in high doses if you just looked in one chemical at a time. So it's common to look at non-parametric approaches. And so you might do something like this. I should say also Y is often not continuous. I'll ignore that. But we have some sort of YI, and you're related to the exposures, the mean of YI to the exposures through some sort of function F, which is often in this literature referred to as a response surface. And we could use the GP or BART or any other sort of non-parametric Bayes regression method in that context. And that's commonly done. But if you do that, then it's really not very useful. So A, you're going to get a pretty crappy estimate of F in the types of sample sizes and P dimensions that we have in these studies. B, it's not going to be very interpretable at all. And so we don't want some sort of black box to predict your health. We would like to understand the effects of these different chemicals, for example, for regulatory purposes. And so we really want to disambiguate the different chemicals and not to just have a black box for prediction. So some existing approaches. So one thing that's really popular in this literature is what's called Bayesian kernel machine regression, which is developed by the group at Harvard, including Brent Kuhl and others. And that's really quite popular. They have a nice package. And what that does is it's basically just a Gaussian process with variable selection, which is something that's been done in other contexts, but they've really done a fantastic job getting this package and getting it popularized in the epidemiology community using these Bayesian tools. So with variable selection, this f of x is a lower dimensional. Maybe now it's only 5-dimensional, but that's still pretty big. And it's maybe really hard to visualize and understand and interpret this 5-dimensional surface. So alternatively, something that you might try is to kind of decompose the function into simpler parts. And so you might say, oh, well, f of x, we might have a linear part, and then we might put in a quadratic part with like a parametric interaction part, which is what Tamara was doing. We might also add an additive part, additive nonlinear parts. We might add in, 2-dimensional interaction surfaces in each predictor. So we could kind of decompose the function in that way, and we could think about variations on that theme. Okay, and so in the parametric case, which is what Tamara was doing, this is called quadratic regression. And there's a big literature from a frequentist perspective. Some of those approaches both Tamara and Sylvia were showing. And so they just have this kind of parametric quadratic term. Okay, and so we had like in the same type of context, but kind of motivated by the environmental health applications. We had a recent JASA paper where we were, also trying to address the kind of computational and the statistical curse of dimensionality, but in a different way, using latent factor models. And so you're thinking this xi vector is high dimensional. What if there's a low dimensional set of factors underlying that high dimensional xi? And then we could put those latent factors instead of the original xi's in the response model. We could both put them in linearly and quadratically. And so then actually you end up getting a much smaller computational burden, which should be like we didn't go into that, and that wasn't our main focus, but perhaps is definitely kind of subquadratic in P. So that would be interesting to explore whether this kind of latent factor modeling trick allows you to kind of never be dealing with this kind of high dimension P squared space, because you could just pick out which the loadings based on the zero structure and the loadings, which interactions are in. And so you don't have to calculate all possible P squared interactions doing this type of thing. That wasn't our focus on computational complexity, but Tamara's cool stuff kind of brought that to mind. Yeah, so that's just that point. What about semi and non-parametric extensions, which is something we're interested in? So dose response, it certainly is nonlinear, but maybe we can pick up some of these things with these kind of quadratic regression models. But what if we want to try to get rid of the linearity? So one thing you could do is you could put in like a linear effect with variable selection. You could put in an additive effect, and then you could put in these kind of pairwise interaction surface effects, and that might be a nice compromise. The model seems like overly parameterized, but it might have good interpretability if you could kind of separate these parts and do variable selection for each piece. So what you could do to address the issue with identifiability is kind of borrow a trick from spatial statistics. Chris Patrick and some other people have some beautiful work on this, where you basically use Gaussian processes, but you do this kind of orthogonal projection trick. And so the additive parts can't live anywhere close to the linear parts, and the interaction surfaces can't live anywhere close to the additive or linear parts, and then you're able to get identifiability in that way. And we have a paper that just came out recently in Annals of Applied Statistics doing this, and it seemed to work pretty well. We weren't looking at like thousands of exposures, anything like that, but be interesting to try to kind of scale up some of these ideas. And we have code for all these things. I'd love if people tried them out, certainly. So some questions and thoughts. So the storage and computational issues raised in Tamara's talk might be even more challenging in the semi-parametric case. I think she mentioned at the end that she's already thinking about that, and there's some sort of directions forward there. You might have to back up P quite a bit to deal with this kind of non-parametric models. I made the point before, if P is much less than N, you're actually kind of screwed, to be honest. So because in the simulation study, you're always like, oh yeah, all our signals are enormous. But in reality, and particularly in these types of studies I work on, biomedical studies, these coefficients aren't all that enormous. And even taking all of the predictors we have, we might only be able to explain 20% of the variability in the health outcome or something like that. So these aren't going to be good as predictive models. We're trying to pick up like low signal coefficients, and we're going to have very low power in this case. And so that's the
David,
kind of, yeah.
Sorry to interrupt, just to give you a heads up, if you can wrap it up so that Tamara
Okay,
can
so we are trying to deal with this. I'll just stop there, and yeah.
Yeah, sorry to do that. It's just that I got a warning
no,
that
it's no problem.
the session will
all.
expire. Okay, so Tamara.
Cool. Yeah, great. Okay, wow. I can't imagine two more perfect discussants, like to have two people who are like so on top of their game statistically and in applications. Like, this is amazing. So let me try to address some of these. Also, I just want to say, wow, so amazing that like you thought so much about this paper. I'm very honored by that, and I think that's like super cool. Okay, let me try to address some of the things that you have brought up. Okay, one, this XYZ lasso, that's amazing that you've done this comparison, and we'll definitely make sure to think about that more going forward. We did not do those comparisons in our original paper, and it's great to have that pointed out to us. We have experienced the same thing you have, at least to the best of my knowledge of your discussion, Sylvia, that the MCMC is really a limiting factor. And so there were two issues that actually we seem to see with the MCMC, which is part of the reason that one, support the Pyro change to map, but also have actually switched to a more map-based inference in the new paper that I just mentioned as well. So one is definitely the issue of speed. And I think that this was an issue that came up in yesterday's session on not interactions, but like finding sparse effects in a data set. But another one also seems to be some challenges just in using MCMC for sparse detection of effects. So we have to be sort of careful, and we have like a little appendix on this in the original paper about saying, okay, well, is the mean sufficiently far in terms of posterior standard deviations from 0? Whereas in MAP, something that we were able to do in the new paper is use more of a spike and slab type prior, and then you can sort of say directly, okay, I have this linear representation of the covariates. Is it associated or not? There's just 0 or non-zero. And so there's sort of an ease of that too. Something that this doesn't get away from though, and seems to be I think a well-appreciated problem by many people already, is that this is a super highly multimodal landscape, and that's very challenging. And so, how do you really know that you're getting a good mode when you're doing this, and how do you make sure that you're finding a good mode? That seems like just fundamentally a challenging problem. I still think that it's a good idea to go from this P squared space to this P space. I mean, if anything, it like reduces the nature and like size of that multimodality problem, but that doesn't get rid of it. And it's certainly one for both MCMC and MAP. And actually, one of the things I'll just bring up that comes up in the MCMC too, in particular when you're doing this like mean plus or minus like 2 standard deviations kind of thing, is that you can have an issue of like 2 modes on either side of 0. And then, what are you doing when you're averaging that? It's you're like averaging isn't like the right thing to be doing, is kind of the issue. And Raj and I had talked about this in terms of like VB too, because that's another thing that people do to scale things up, but it doesn't, VB has this tendency to pick out modes, but like it's supposed to be approximating the full posterior mean too. And so anyway, there's some issues there. And so this is partly why we've gone to MAP. I will say that I still think that even using MAP, there are a lot of really nice things about the Bayesian framework for this. You know, one potential benefit, and I love this sensitivity analysis, and I think we're gonna have to think about this too. I mean, there's like similarly a hyperparameter even when you do something like spike and slab, and so we'll have to look into that hyperparameter. One thing that's really interesting here is, so even when you're doing MAP, you're getting, I think, a benefit from at least the sort of graphical models framework in Bayesian inference, because you're able to use these priors like horseshoe and spike and slab that are closer to like basically an L0 penalty than an L1 penalty. And so that at least gives me hope that, like, you know, even if we do, or especially if we're careful about the sensitivity analysis, but even when we do it, that there really are some benefits to this framework over Lasso, and they're not just because we've happened to have chosen like the trade-off to be different. And so I think there's something really interesting there. I love that you pointed out like not only a third order interactions example, but did it. That's so cool. I hope we'll have a chance. I hope all of us will have chances to chat more after this, because
yeah, just to interrupt again, we're supposed to be done.
Oh, no.
I think in 2 minutes, this will self-destroy. So before we go, I just want to thank you for a wonderful talk, and thanks to the discussants. But you can continue talking.
Okay, well, thank you so much. If I get nothing else across, that'll be it. Let me try to like hit some more things. And I'm sorry that this means like necessarily I'm getting to David's stuff a little later. Okay, so another one is, yeah, this runtime that you saw, the 11 hours in a real life data set for MCMC, we totally agree that this, again, part of the reason we've gone to MAP. So in MAP now, I think we've gone up to possibly 7,000 or 14,000 covariates pretty successfully in reasonable runtimes. And so, but I think it's still really an open question if we want to go to like UK biobank sizes, you know, how to really deal with this and make sure that it really does scale appropriately, that it's something that doesn't truly take days to run. Let's see. Is there still a gap for going beyond two-way? I mean, I guess this is part of the reason that we really should do three-way things and try them out. And we weren't so motivated to do that before, but now we're very motivated by this great example. I hope and suspect that actually doing that with the new work in the functional ANOVA work that we've just put out will be easier and that it'll be more automatic. But I think the reality is like we have to do it. And like I feel like every time you actually try doing something in statistics, it turns out there's something that needs to be like filled in or like some gaps. So so I don't want to promise too much, but I think we would love to like look at that more and hopefully fill in whatever gap does exist. David's discussion, I think, raised some really interesting questions about, I feel like there's a really interesting question here about what are the fundamental limits of what we can actually do? You know, like somehow, I feel like this comes up in a lot of areas that like we push to do things faster, to do things more accurately, but at some point, presumably there's like a statistical limit about what we can expect to actually be able to do. And it'd be interesting, I think, to better understand that, not to say that I have a solution to that. Some of my things I said already are also responding to David's point. So like, I think like the benefits here are getting this closer to the L0 penalty. The nonlinearity of dose response curves is super interesting and cool, and hopefully a motivation for doing some of this functional ANOVA stuff that we've been doing. And I'd love to chat more about that too. What we've been approaching, how we've been approaching this is like the equation that you put up, David, but without that leading linear term. So it's sort of saying, like from a functional ANOVA standpoint, how we deal with identifiability is saying, okay, you know, you can say that there are sums. Your total function is a sum of functions that depend only on one covariate, and then a sum of functions that depend only on two covariates, and so on. And then in particular, the two covariates should be such that they cannot be expressed as one covariate, or the one covariate cannot be expressed as a constant. And so what we've been doing is really building on like this existing functional ANOVA literature. I should say, like, we're not, you know, creating the functional ANOVA, but I think that we can actually get some pretty good scalability within this framework by using basically these like non-parametric basis functions and getting these very general functions that way. So I know we just put this paper up in the archive, but I hope I can get you all to take a look. And I'm sure we'll be revising it based on some of this great feedback here. In terms of storage and computation, I think that, well, okay, so there's going to be some trade-offs here. So one is definitely choosing the appropriate sort of bases to be working in. And I think that's going to relate to these things like the sensitivity analyses and so on. But we do find that for some reasonable bases, like B-splines with an appropriate number of knots, that we can get seemingly pretty good storage and computation, and that if anything, like the MCMC was really holding us back. And so hopefully we can still see that going forward. And I think I rushed through everything, but I
Yeah.
hope I've addressed most of the comments.
Yeah, there is also a question in the Q&A,
Oh,
but in the
great.
interest of time, I think we need to
Oh,
definitely
gotcha.
end.
Okay, I will try to
but
answer
maybe
this
you can
question
connect.
online then in the Whova chat.
Mhm. Okay, well,
Thanks, Suzanne,
yeah,
that was
so
fun.
we have, thank you so much. And yeah, hope to see you at ISBA next year.
Totally. Great. Thank you, everybody. Thanks so
better.
much.
Bye. Bye, everyone.